Hello everyone, in today's video we are going to learn about acid-fast staining or Zeihl-Neelsen staining. This type of staining is mainly used to identify acid-fast organisms, mainly Mycobacteria. It is named after two German doctors who modified the stain. Bacteriologist Franz Zeihl and pathologist Friedrich Nielsen. So this method was originally developed by Paul Ehrlich, and the modified version is known as the Zeihl-Neelsen staining. It is most commonly used to identify the members of Mycobacterium, especially the Mycobacterium tuberculosis and Mycobacterium leprae, the pathogens which are responsible for tuberculosis and leprosy, respectively. These organisms are difficult to stain by ordinary staining methods due to presence of high lipid content, that is mycolic acid, in their cell wall. So these organisms have cell walls containing lipids constructed from mycolic acids, which is a group of branched chain hydroxyl fatty acids that prevent dyes readily binding to the cells, and hence staining, normal kind of staining, is difficult. So this special method of acid staining is used to identify these organisms. In this method we use heat and phenol to drive the basic fuchsin into the cells. Once the basic fuchsin has penetrated, Mycobacterium tuberculosis and Mycobacterium leprae are not easily decolorized by acidified alcohol and thus are said to be acid-fast, whereas the non-acid-fast bacteria are easily decolorized by acid alcohol and are stained by methylene blue counterstain. The reagents used are Carbolfuchsin solution, acid alcohol solution, methylene blue counterstain. Let's move on to the procedure next. Prepare a smear of the specimen using an inoculation loop on a clean glass slide. Just a thin uniform portion is needed. Next step is to air dry and heat fix the smear. After heat fixing, flood the smear with freshly filtered Carbolfuchsin. Then heat gently until the steam rises. Continue to heat for 5 minutes so that steam is seen but without boiling. One point to note here is that we should not allow the slide to dry. So to prevent that, we should add more stain from time to time. Allow the slide to cool off and then wash it under low stream of running water. So next step is the application of acid alcohol, which is the decolorizer. This step we should be able to identify the acid-fast, as acid-fast organisms are not easily decolorized, whereas non-acid-fast organisms are easily decolorized. So, cover the slide with acid alcohol solution for 3 minutes. The slide is then washed again under running tap water and drained. Now cover the slide with methylene blue stain and leave it for 2 minutes. So the methylene blue is the counterstain here. After this, wash with tap water, blow dry or air dry the slide and observe under microscope in the oil immersion objective so that we can observe the organisms under high resolving power clearly. So the result we obtain here is that the acid-fast organisms retain the primary stain, Carbolfuchsin, after the application of strong acid and appear red, whereas The non-acid-fast organisms do not retain the primary stain and are counterstained by methylene blue and appear blue color. In this figure, you can clearly see the difference, how acid-fast and non-acid-fast are stained differently. At first, both of them are stained by Carbolfuchsin. All appear to be reddish color. Then, after applying the strong acid alcohol solution, which is a decolorizer, meaning that they will remove the stain, the acid-fast organisms retain their red color, whereas the non-acid-fast become colorless. Then the non-acid-fast are counterstained with methylene blue and appear in blue color, whereas the acid-fast still remain red.